Yes, thanks.
Yep. Susan and I are going to be presenting together today, and I'm Chris Havran.
Hi, Susan.
Susan, and switch, please. We're really happy to be able to talk about some collaborative research that we worked on some time ago that fits today's workshop theme, especially seeing how molecular techniques may help to resolve some uncertainty among relationships in Hawaiian taxa. Susan, I'm going to pass it to you. Switch.
Thanks, Chris. I just wanted to take a moment. Thank you to James Kwan for the lovely introduction this morning. This quote is similar. I wanted to just say that I had the privilege to spend a lot of time in the field with this guy, Joel Lau, and he taught me a lot about how to observe plants in the wild here, and also Steve Proman. You know, I think it's really telling that I have no pictures of their faces. It's just their backs and them actually obsessively looking at plants and the habitat. From this perspective, I learned conservation through their eyes, which groups are more prone to morphological variation or hybridization. When they thought something was interesting, rather than just observing an aberrant individual or even a reaction to an abiotic factor such as too much light. They could, Joel especially can and still does quote Scottsburg, and I don't necessarily have that ability, but I definitely ask a lot of questions and have been interested in the history of collections of various species and their distribution and how they're different from their congeners. What used to be collected in what places and what was common and now isn't. And this continues to influence how I look at morphological entity, its geographical distribution in relation to other things that have previously been described. And but the base of it is always comes back to how can we conserve these various forms. Next slide. I'll just briefly say that I was taken to the Oahu population of this Viola kauaiense, and you know, it's extremely different than what is described as far as the plants on Kauai, and we all just talked about it. Joel had mentioned it had previously been described by St. John, but wasn't currently recognized, and so. But we thought, you know, hey, it's so distinct and extremely small population. So I continued to try and visit and make collections, but I was asked by people from my supervisors from PEP, DOFA, and Fish and Wildlife Service, why am I working on this? It's relatively abundant on Kauai. So I decided to bug my friend Chris with all of my questions and ask him to look into it. And I may have asked him more than once. Chris.
Yeah, thank you, Susan. And next slide, please. Yeah, Susan had contacted me several times a couple years ago. I did my dissertation on the ecology and evolution of the Hawaiian viola. And Viola kauaiensis, as Susan mentioned, is really prevalent on Kauai, especially in Kokei State Park. If you've seen this before out there, it's a creeping rhizomatous herb. It's on the edge of the boardwalk and in the margins of the cloud forests. It's unique in that it's the only Hawaiian viola with an herbaceous growth form. Most of the other Hawaiian viola out there in Hawaii are woody subshrubs. It's also unique in that it produces chasmogamous or open flowers, like you can see here, and then cleistogamous or closed flowers. Here in North Carolina, I'm used to the violets with their nice chasmogamous flowers early in the season and cleistogamous later to facilitate selfing in the absence of insect pollinators. That likely helped these violets get a foothold in Hawaii millions of years ago when they were first established. Switch, please. But in 1938, Edward Hosaka had discovered a new population of Viola kauaiensis on Oahu in the Ko'olau Range. St. John had named this Viola Hosaka, and the manual treated this as possibly another taxon of Viola kauaiensis. But taxonomic work had not been done on this since St. John published this as a new taxon. Next slide, please. The earliest phylogeny of the Hawaiian viola was conducted by Ballard and Seitzma in 2000. And even though this doesn't include all Hawaiian taxa that we know today that are in the genus Viola, the population on Oahu is what was still represented here as Hosakai, which forms a nice, well-supported relationship with Kauaiensis. So even in this early phylogeny, there was a really tight relationship between these two inter-island populations of these herbaceous violets. Next slide, please. For my dissertation in 2009, we published this revised phylogeny of the Hawaiian viola, including inter-island populations of every species, expanding the size of the phylogeny a bit. Those two populations of Kauaiensis on Oahu and Kauai still form that nice tight relationship, though we recognize them by the name, the manual at the time, Kauaiensis on Oahu. The next slide, please. This same phylogeny had been used to evaluate a previously uncertain relationship. In around 2012, Hank Oppenheimer had contacted me about a new discovery of viola that he had uncovered on Maui at Helu. So we sequenced the ITS region of that material, incorporated it into the previous phylogeny, and saw that it formed a polytomy with Viola oahuensis on Oahu and then Viola lanaiensis on Maui. So that sequence was not enough to show that it was monophyletic with Viola lanaiensis on Lanai. So we followed that up with a morphometric analysis shown in C there of herbarium material of Lanaiensis and Oahuensis and newly collected material by Hank. Along that canonical variant 1 there, you see that line of dots going up and down the y-axis. That represents all of the material of Lanaiensis and then material from Lanai, or that Hank had previously seen. So we were able to start with the phylogeny and then move on to a multivariate morphological analysis to demonstrate that that population on Lanai did represent Lanaiensis. I proposed to Susan that we do something similar with the material from Oahu. Next slide, please. So Susan and I, along with Joel Lau and Clyde Amata, made several visits into the Ku'uaus in 2013 to attempt to rediscover previous populations of this entity on Oahu to make some new collections. The original type locality in the Ku'uaus by Hosaka is currently unknown. However, in 1986, John Obata had seen this population, or had seen this entity along the Ku'uau Summit Trail. He shared information about this population with Clyde Amata in 95, who revisited this population and didn't find any. We revisited it again in 2013 and again didn't find anything. So that population is presumed extinct. However, a population that Susan had visited before along the Po'omoho Trail did support 4 small subpopulations of this violet. Next slide, please. This population seen there in image A is really different than what you would see on Kauai. Here on Oahu, this violet is on relatively steep cliffs. It's growing right out of moss in a really exposed environment. And on Kauai, you would expect it on flatter, more not submerged, but flatter, wetter substrate. Really different here on Oahu. We found 50 individuals and made 2 collections for the Bishop Museum. We didn't want to disturb any other individuals, but we wanted to get an idea of the morphology of the population. So we ran a transect along the ridge and measured leaf characteristics of as many individuals as we could, just to get some idea of the range of morphology of these violets. The leaves are very, very tiny, as you can see here, much smaller than what you would normally see out on Kauai. And as we were visiting this population, we only observed cleistogamous flowers. So no open chasmogamous flowers were seen. And so we were only able to collect something that was presumably cleistogamous. Next slide, please. Since this population, or since the entity on Oahu had already been sequenced, we conducted a morphometric analysis. This is a principal components analysis here. All Oahu populations are these little black squares they kind of cluster together with three little individuals, those little black triangles, that represents something that St. John had called Viola van Royi at Waialeale. Everything else in the open circles and squares here represents Viola kwaiensis. We incorporated some, at the time, current Waialeale specimens into the analysis, which clustered with all the other kwaiensis specimens here. And then conversations we had with Steve Perlman indicated that the site location for this Van Royi entity named by St. John is sympatric with Viola kwaiensis, as it's known. So because the population of our Viola from Oahu here, little black squares, was just on the margin of the morphological range of kwaiensis as a whole, and because it was geographically isolated, we argue that it should represent its own distinct taxon. Next figure, please. So the taxonomic treatment we made of this population was Viola kwaiensis variety Hosaka. Its habitat is on, again, a moderate to steeply sloping surface, growing directly out of soil or moss. It has a really limited range of morphological variation relative to those Kauai populations. Some leaves from the Viola kwaiensis on Kauai can get really tiny, but as a whole, the population shows a broad range of morphological variation. And this is really closely related to Viola kwaiensis on Kauai. So there's only 4 nucleotide differences between these inter-island varieties. Next slide, please. So Susan has been able to conduct some conservation activities related to these finds. Susan?
Thanks, Chris. So yeah, I guess now that it was recognized, I felt more justified in spending a lot of time on this species. I should say that the Hawaiian name recorded is Nani Waialeale, which would be a Kauai name, I think, also Pohehiva. So yeah, the population ever since I'd visited had been declining and had been described to me as being much, much larger by Steve and Joel. So when I counted, it was in the 80s, and then now we're currently seeing 30 or less individuals. So we have made collections from about 1/3 of those. They're hard to catch in fruit. We're also noticing that the slope is being exposed to a lot more hot, dry, cloudless days, and the moss that they live in is drying out. So I did do a translocation of an individual from the dried moss to a wetter patch of bryophytes, as that picture shows in the middle there. And then it also shows some of the seed collections and also plants in micropropagation. So from the collections, we've put a few individuals into micropropagation, and then from there, the lab was able to create many, many individuals. Of course, they're clones, but this is really good for us because it allows us to, experiment with the outplanting. So this year we were able to plant 45 individuals, which is much greater than all of the wild individuals combined. And, we're hoping to find that, that they'll do well in this newer, wetter site, which is just a couple 100 yards away from the wild site. And yeah, I think the story is ongoing. So the next slide is our last slide. And, oh, go ahead, Chris.
Yeah, so we just wanted to reiterate how previously established phylogenies can inform these conservation efforts. They can provide a springboard to further morphological studies to help resolve uncertain relationships. One thing I wanted to bring up here is this population on Oahu, as far as we can tell, is self-fertile. It's cleistogamous. Susan, you've not seen any chasmogamous flowers. The type collection does have some open chasmogamous flowers on it, but I don't believe any have been seen in quite a long time. So I just want to know what type of genetic resources we have out there that can help us conserve these organisms that appear to be completely selfing. That's beyond the scope of this presentation, just something that I was thinking about for the context of later discussions. One more slide to wrap things up.
That was for me. This lyrics from the song have been in my head a lot lately. everything that we do as far as our conservation work here in Hawaii, are for this place, this story, these species, this place, this land, and we have so much aloha for it. And I just wanted to thank Chris for having that aloha too. That's it.
Thank you, Susan, and thank you everybody who has organized this workshop to allow us to share ideas and learn how to help. Thank you.